An emerging view of the diversity, ecology, and function of archaea in alkaline hydrothermal environments. Abstract. The described diversity within the domain Archaea has recently expanded due to advances in sequencing technologies, but many habitats that likely harbor novel lineages of Archaea remain understudied. Knowledge of Archaea within natural and engineered hydrothermal systems, such as hot springs and engineered subsurface habitats, has been steadily increasing, but the majority of the work has focused on Archaea living in acidic circumneutral environments. The environmental pressures exerted by the combination of high temperatures and high pH likely select for divergent communities and distinct metabolic pathways from those observed in acidic or circumneutral systems. In this review, we examine what is currently known about the Archaea found in thermoalkaline environments, focusing on the detection of novel lineages and knowledge of the ecology, metabolic pathways, and functions of these populations and communities. We also discuss the potential of emerging multi-omic approaches, including proteomics and metabolomics, to enhance our understanding of the Archaea within extreme thermoalkaline systems. Background. Technological advances have led to a wide array of high-throughput methods for documenting the abundance, diversity, and functional potential of the domain Archaea. Despite this progress, archaeal communities in key environments remain poorly understood. Sites characterized by high temperatures coupled with pH conditions that range from near neutral pHs of 6.5 to 7.5 pH units to alkaline with a pH greater than that of 7.5 pH units represent environments known to host novel lineages within the Archaea and have been rich resource for thermostable alkaline-tolerant enzymes for biotechnology. Archaea that can survive under the polyextreme conditions of both elevated temperature and high pH conditions have unique functional pathways, which in turn represent novel biochemical functions. In this review, we examine patterns of microbial diversity within thermal and circumneutral and alkaline ecosystems. In an effort to better understand the ecology of the Archaea, we included studies that targeted the total microbial communities in these sites, the prokaryotes, bacteria, and Archaea, as well as both natural hot springs, serpentinizing marine vents, and engineered bioreactors, drilled wells, systems. Further, to examine the differences among low and high pH sites, we included studies conducted across gradients of pH and temperature to identify abiotic drivers of community composition and function. Using currently available literature, we 1. Explore the ecology of archaeal communities, including the likely environmental drivers of community assembly and coexistence. 2. Review metabolic and functional pathways found in archaeal taxa in thermoalkaline systems, including how functional diversity responds to changing environmental conditions. And 3. Outline the progress made in identifying novel lineages, including the use of single-cell genomics, metagenomics, assembly and metagenome-informed culturing methods. Based on our assessment of the ecological characteristics required to survive in thermoalkaline environments, we postulate that the ecological systems of host still uncharacterized archaeal lineages with novel functionalities and metabolic adaptations. However, we are still in the early stages of understanding how thermoalkaline archaeal species adapt and survive. Undersampled thermoalkaline systems. Thermoalkaline environments are found in terrestrial and marine ecosystems, including liquid phase-influenced hot springs and serpentine vents formed when ultramafic rock is exposed to water. The microbes that inhabit hot springs are perhaps the best studied of the high pH environments and are found globally, but molecular surveys of microbial communities have been focused on a limited number of sites. Of the 400 documented hot springs in India, 28 have been sampled using either culture-based or high-throughput sequencing methods. Similarly, in YNP, a subset of alkaline hot springs have been sampled extensively, such as octopus and mushroom springs. Two regions in YNP have a large number of thermoalkaline springs, including Heart Lake Geyser Basin and Lower Geyser Basin, which are often sampled as part of large-scale surveys that span temperature and pH gradients found within the park. The Uzan caldera in Russia host springs with a wide range of pH, ranging from 3.1 to 9.8 pH units, but most hot springs sampled in Kamchatka are acidic to circumneutral, with pHs of 3.1 units to 7.0 units, or are locations with pH conditions that span acidic to alkaline. Sites with geothermal activity in Central and South America, such as those in Costa Rica, Peru, and Argentina, also represent potential reservoirs for taxonomic novelty in the Archaea, but to date, studies have focused on acidic and circumneutral sites, and the density of higher pH springs is unclear. In addition, there are geothermal regions that are just beginning to be explored, such as the estimated 240 hot springs in Algeria. In marine systems, serpentinizing thermal vents support archaeal communities dominated by methanogens, but a smaller number of alkaline sites, such as the Lost City hydrothermal field, have been examined relative to cold or acidic marine sites. Clearly, there is more to be discovered on the diversity and function of archaea in thermoalkaline systems. Polyextremophile archaea, challenges of high pH and temperature. Archaea that live in thermoalkaline environments are polyextremophiles, able to withstand both high temperatures and high pH. Adaptations to temperature include modifications to the cellular membrane, changes to DNA repair pathways, and protein stabilization. Organisms that survive and thrive in extreme pH conditions are generally maintain internal pH that varies drastically from their environments. Under alkaline conditions, the best documented mechanism for maintenance of near-neutral internal pH is the use of antiporters, sodium hydrogen, and potassium hydrogen, that exchanges ions for protons. Under the polyextreme conditions of both high pH and temperature, the trade-off between investment into tolerance and metabolic pathways can alter patterns of growth and activity. For example, the alkalophile Natrinobiforma cellulositropha, which could grow up to a pH of 9.9 units at low temperature, could only grow at the lowest pH values included in the study, 8.5 when temperatures exceed 43 degrees Celsius. Ecology and diversity of archaea in alkaline hydrothermal sites. Biogeography of archaea. Biogeography, the study of the distribution of species and communities over geographic, environmental, or temporal gradients, can be used to understand the mechanisms determining microbial community abundance, diversity, and composition. By identifying the range of conditions in which a particular taxon inhabits, studies conducted across broad geographic and environmental gradients provide insights into whether certain archaeal taxa occupy specific niches, thus increasing our understanding of environmental factors which limit archaeal species distribution. Large-scale studies, such as those conducted across 925 hot springs in New Zealand and 118 hot springs in the Tibetan Plateau, can begin to identify the environmental breadth of given taxonomic groups of Archaea, as well as the dispersal limitations that may constrain their geographic ranges. Comparisons of archaeal operational taxonomic units, or OTUs for short, detected in geothermal hot springs that varied in both their temperature, 7.5 degrees Celsius to 99 degrees Celsius, and pH 1.8 units to 9.0 units across Canada and New Zealand, found that the sequences detected had close matches to those found from geothermal sites in Japan, Thailand, Tibet, and YNP. This indicates that archaeal taxa found in hot springs have broad geographic ranges, suggesting dispersal limitation is not a predominant factor driving archaeal distribution in alkaline hydrothermal systems. Similarly, based on metagenomic sequencing, a number of uncultivated Igarchaeota, Coarchaeota, and Nanoarchaeota, Archaeophylla, were distributed across 4 continents. Large-scale studies have revealed that functional classifications based on the presence of organisms under specific pH and temperature conditions, such as surveys that attempt to categorize lineages as acidophilic or thermophilic, may reflect less robust sampling rather than true physiological constraints. For example, recent genomic surveys of several alkaline springs documented archaeal taxa that were previously thought to be localized only to acidic systems. In one study, amplicon and metagenome surveys from the moderately alkaline pH of 8 hot spring in Malaysia identified sequences most closely related to Acidiloprofundum bunyi and Sulfolobus acidocaldarius, taxa considered to be acidophiles. As the number of environmental genomic surveys expand, so does our ability to identify the geographic and environmental ranges of the archaeal taxonomic groups. This same trend holds true for high pH geothermal habitats, where a recent 16S rRNA survey conducted an alkaline pH of roughly 8.7 units hot spring in Russia detected an environmental sequence identified as a species in the genus Forvidococcus that was nearly identical to a phylotype originally detected in a circumneutral pH of 6.7 to 7.6 hot spring in the YNP. Additional studies conducted across both large spatial gradients, particularly those that target more than phylogenetic markers, could provide additional insights into patterns of evolution and adaptation across both spatial and environmental gradients. Figure 1. Archaeal communities in circumneutral and alkaline pH terrestrial geothermal systems across the globe. Hot springs have been documented in multiple countries, solid gray. A small number of regions, including China, New Zealand, and the USA, primarily YNP, have been more robustly sampled using current omic approaches such as metabarcoding and shotgun metagenomics circles. Other geothermal sites have documented thermoalkaline sites, but have not sampled using modern methods, diamonds. And others have not been described in terms of temperature and pH ranges, triangles. Colors of symbols correspond to pH of the hot spring sample, scaling from circumneutral to alkaline. Circle size corresponds to the number of circumneutral to alkaline hot springs sampled. Data was drawn from published papers as well as sequence data available in public databases, including GenBank, NCBI. Survey of thermoalkaline sites. In order to understand the roles of archaea in these systems, a better understanding of the range of diversity present is needed. To gain a more holistic understanding of the archaeal diversity present in thermoalkaline sites, and to examine the phylogenetic distribution of environmental sequences from thermoalkaline sites, we downloaded raw 16S rRNA sequence data from NCBI for hot spring sites from China, YNP, Russia, India, Japan, and Romania. We also included additional sequences generated from sediment slurry collected in YNP, including the 5 sisters, and sites previously surveyed in Heart Lake Geyser Basin. In order to compare among sites, we limited our analyses to studies that targeted the V3 or V3-V4 region of the 16S using paired end sequencing on the Illumina MI sequence platform. FASTQ files were filtered using usearch with an expected value of 0.5, and operational taxonomic units, OTUs, were delineated using UNOISE2, UNOISE2. Environmental OTUs were aligned to full-length and near full-length 16S sequences from Archaea downloaded from NCBI and the GTDB. Sequences were aligned using MAFT, MAFFT, and a maximum likelihood phylogeny was inferred using RAXML with the GTR plus gamma model. The phylogenetic and taxonomic classifications of Archaea continued to shift, and so we assigned taxonomic identification of reference sequences based on Baker et al. 2020. For example, based on NCBI taxonomy, the phylum Archaeota is part of the superphylum Archaeota, but is placed with DPANN in later classifications. The phylogeny was annotated using the interactive tree of life. Although universal 16S primers may not detect all members of the Archaea, later iterations of commonly used primers, such as the 515F806R Earth microbiome primers, have been modified to reflect additional diversity. The phylogenetic placement of the several archaeal phyla from publicly available sequences was not monophyletic, possibly due to the use of 16S rRNA sequences from MAGs and SAGs, but from this analysis several patterns emerged. First, there were large numbers of OTUs, operational taxonomic units, that were closely related to phyla in the DPAN superphylum, particularly the Woas Archaeota and Pachyarchaeota, as well as the TAC phylum, Bathyarchaeota. To date, none of these groups have cultured representative, and thus, these sites could be targets for culturing efforts of these groups. Second, the number of the OTUs from the known methanogenic groups was comparatively low. However, given the expansion of methanogenic pathways in taxa outside of Archaeota, methanogenesis may be undertaken by other groups. Finally, only a small number of OTUs were placed with members of the Asgard superphylum, which could be due to the lower number of reference sequences available, but could also be indicative of lower abundance of this group in alkaline sites. Figure 2. A phylogenetic tree based on full-length and near full-length archaeal sequences from NCBI and the GTDB, and environmental OTUs generated from publicly available MI sequence data across a range of locations, including YNP, China, India, Japan, and Russia, and drilled wells in Romania. Reference sequences are colored according to their current phylum level classification. DPAN, blue. Archaea, green. Asgard, red. TAC, orange. Environmental sequences are not colored, likely due to the use of the 16S sequences from MAG or SAG. The tree is not monophyletic, particularly for the DPAN superphylum, where the number of cultured representatives is minimal. Environmental drivers of archaeal abundance and diversity. Temperature. Temperature is known to be a strong driver of archaeal abundance in hydrothermal environments. With high temperature, greater than 80 degrees Celsius, conditions favoring the formation of ecological systems with higher proportions of Archaea. For example, a comparison of several sites in New Zealand and Canada that represented a gradient in both pH, roughly 2 to 9 pH units, and temperature, roughly 10 to 100 degrees Celsius, found that the relative abundance of the archaeal phylum Cranarchaeota increased at higher temperatures, from 1% at 0 degrees Celsius to 20 degrees Celsius, to nearly 50% at 80 degrees Celsius to 100 degrees Celsius. Correspondingly, a qPCR-based analysis of 3 abandoned geothermal oil wells in Romania with high levels of bicarbonate, HCO3-, and temperatures of 32 degrees Celsius, 49 degrees Celsius, and 65 degrees Celsius, noted that the relative abundance of Archaea was 36% of the total community in the 65 degrees Celsius well, but less than 1% at cooler temperatures. When relative abundance was examined using qPCR, the relative abundance of Archaea increased from 45% in the highest temperature well and less than 2% in the 32 degrees Celsius and 49 degrees Celsius well. The relative abundance of Archaea is nearly always maximized at higher temperatures, but diversity patterns are less clear. Ribosomal genes detected in shotgun metagenome surveys generally indicate that Archaea constitute a lower proportion of the microbial diversity compared to bacteria, regardless of temperature, and this pattern is also generally found in targeted surveys performed alongside shotgun sequencing. 2 studies that examine changes in microbial across broad temperature gradients found curvilinear patterns where diversity peaked at mesic temperatures, roughly 21 degrees Celsius, and plateaued above 65 degrees Celsius. A pattern also observed at the Lost City vent. Archaeo-focused studies have revealed less consistent patterns. In Iceland, a comparative study of Archaea across 4 circumneutral hot springs with identical pHs of 6.5 pH units found the highest diversity of 88 degrees Celsius, but the lowest at 90 degrees Celsius, with intermediate values observed at lower temperatures, 65 degrees Celsius and 67 degrees Celsius, suggesting an abrupt threshold at extreme temperatures. In contrast, Song et al. 2010 found that moderate temperature springs hosted the highest levels of archaeal diversity, but were dominated by members of a single phylum, and Crenoarchaeota, members of a single phylum, the Crenoarchaeota. In Romanian wells, the number of operational taxonomic units, OTUs, was similar across different temperature wells, but a single genus, Methanobacteria, dominated at higher temperatures, resulting in shifts in community composition due to the high dominance of punitive methanogens at higher temperatures. Although function is not always predicted by phylogenetic diversity, a better understanding of the phylogenetic breadth of communities could provide insight into the potential functional diversity across temperature and pH gradients. Environmental drivers of archaeal abundance diversity. pH. As shown in soil across numerous ecosystems, pH can be a driver of microbial abundance in hydrothermal systems. In focus studies of alkaline springs, Archaea rarely exceed 10% of the overall microbial population abundance. And generally, 16S rRNA gene surveys have concluded that Archaea are more abundant in low pH hot springs in comparison to their alkaline counterparts. This pattern is consistent across regional geographic gradients as well, as Power et al. In 2018 focused that Archaea exceeded 1% relative abundance only in acidic to circumneutral springs. This was corroborated by a study conducted in Costa Rica that did not detect Archaea in any hot spring with a pH above 6. While the abundance of Archaea may be lower in alkaline hot spring systems relative to their acidic counterparts, as with the effects of temperature, there is some evidence that archaeal diversity increases at higher pHs. 16S rRNA gene sequences analyzed from one acidic pH of 3.6 units and 3 alkaline pH of 7.5 to 9.3 geothermal hot springs in China found that archaeal phylogenetic diversity increased as a function of elevated pH. In this system, the acidic spring was dominated by a single archaeal lineage, Sulfolobales, while 3 alkaline springs supported a broad phylogenetic range of archaeal populations, including the Disulfuro-Coxales, Eukaryoarchaeota, and Coarchaeota, along with several other unclassified archaeal phyla. These findings come with a caveat. However, as the acidic springs also had the highest temperature of 96.3 degrees Celsius, while the alkaline springs ranged from 48.7 to 88.3 degrees Celsius. Further, a separate comparative study of 38 hot springs across China found that over a temperature range spanning from 55.1 degrees Celsius to 93.6 degrees Celsius, archaeal taxonomic diversity decreased with decreasing pH. pH range from 2.5 units to 9.4 units. As seen in other studies, the lower diversity was driven by the high dominance of the Sulfolobales in low pH springs, while numerous circumneutral to alkaline pH greater than 6 units hydrothermal sites hosted a mixture of Disulfuro-Coxales and Thermoproteales. The study also suggested that there are interactive effects of temperature and pH on archaeal diversity, where at lower temperatures, Euryarchaeota and Thaumarchaeota archaeal lineages dominated in both alkaline and acidic habitats. The pattern of decreasing diversity with decreasing pH reported by Hu et al. In 2013 was subsequently confirmed in a phylochip analysis of a subset of these sites. A comparative study of two high-temperature springs, 51 degrees Celsius and pH of 3, and 93 degrees Celsius with a pH of 8, in Azores Islands off of Portugal, based on pyrosequencing and domain-specific FISH probes, found that a single genus in the Archaea thermoplasma comprised nearly 90% of the archaeal community in the acidic spring, while a broader range of genera including Bacillum, Sulfophobacoxus, and Desulfurococcus in the phylum Crenarchaea were detected in the alkaline spring. Comparisons between acidic and non-acidic hot springs in China also found that archaeal communities were simplified in acidic springs, hosting a restricted range of phyla. Together, these studies indicate that the phylogenetic breadth of archaea in alkaline springs exceeds that of acidic springs, potentially indicating a larger range of functional niches in these systems. Studies using primer-independent sequencing techniques have found similar patterns to those that employ targeted sequence approaches. A shotgun metagenome study in thermophiles from a variety of hydrothermal environments across 4 continents found that genera and the uncultivated archaea phyla Igarchaea, Corarchaea, Nanoarchaea were widely distributed in geothermal sites ranging from pHs of 3.4 to 9.3 units, although the relative abundance varied among the sites. For example, the obligate endosymbiont Nanoarchaea was present at greater than 1% abundance in the 4 acidic habitats with pHs of 2.9 to 4.4 units included in the analyses, but was detected at less than 0.1% relative abundance in 3 alkaline pHs of 6.4 to 8.0 YNP springs, suggesting that pH likely acts as a secondary selection filter relative to temperature. The patterns observed for Nanoarchaea, which is an extremely small obligate archaeal symbiont, can also be linked to the distribution of their host organism, which comprise members of either the Sulfolobales or the Acidobales taxonomic orders, and tend to be more abundant in acidic springs. This body of work suggests that although the relative abundance of archaea in alkaline hot springs is lower than that of acidic geothermal environments, the phylogenetic and potentially the functional diversity of archaea may be higher in alkaline systems. Further work is needed to elucidate the effects of temperature and pH in driving patterns of archaeal abundance and diversity, especially in alkaline thermal systems. Based on current studies, it seems likely that these 2 critical factors interact to drive archaeal community patterns. Additional investigations, including broad-scale global studies and meta-analyses, would likely be beneficial in providing further insights. Outside of the strong selective pressure of temperature and pH, other factors such as organic carbon inputs, sulfide concentrations, salinity, and or dissolved oxygen are known to be important drivers of diversity and abundance in other hydrothermal systems. They may exert significant selection pressures on archaeal communities and thermoalkaline environments. Insights into the drivers of archaeal community composition can be gained by identifying the metabolic pathways used across a range of environmental conditions. Functional roles of archaea. Metabolic pathways of archaea in thermoalkaline systems. Temperature and pH are commonly examined as potential drivers of microbial community assembly and are often closely linked to both taxonomic and functional differences among hot springs, but factors outside of these parameters also play critical roles in driving community assembly. Archaea play critical roles in global biogeochemical cycles and are particularly important in the global nitrogen, sulfur, and carbon cycles through ammonia oxidation, sulfate reduction, and methanogenesis, respectively. As a result, in addition to temperature and pH, available energy sources play an important role in driving both taxonomic and functional composition, and the metabolic pathways of organisms found in particular sites can often be predicted based on the geochemical makeup of the springs. In general, energy yields follow predictable paths based on available electron donors and acceptors, but metabolic reactions favored under one set of geochemical conditions are not energy sources at other conditions. In general, redox reactions that are more energetically favorable will be preferentially utilized by microbes, and detailed geochemical analyses coupled with thermodynamic modeling can be used to predict the most likely redox-coupled reactions, which can in turn be linked to the functional pathways of the microbiome. These models do not account for biotic interactions such as syntrophy and competition, and may be difficult to apply to populations with metabolic flexibility in carbon and energy metabolism, and mixotrophy observed in alkaline microbial communities. Further cross-comparisons among studies can be limited as high-resolution measures of geochemistry represent a significant monetary investment, and they are often not completed, particularly when a large number of springs are included in the study. Carbon cycling, heterotrophic and autotrophic pathways. Archaea utilize metabolic pathways that diverge from those of known bacteria. In extreme hydrothermal systems, archaea are theorized to drive anaerobic carbon cycling through fermentation-based metabolisms. Heterotrophic archaea have central carbon metabolic pathways that are distinct, including modified variants of sugar degradation pathways found in bacteria. Archaeal utilization of organic carbon depends upon inputs into the system. Genome comparisons of archaeophyla using the database of carbohydrate-active enzymes, or CASzymes, are found large differences in the number of type of CASzymes detected, with particularly high numbers of GH in the newly described TACK phylum, Brachyarchaeota. There is evidence that some archaeal taxa in the Bathyarchaeota utilize lignin. While lignin deconstruction is energetically costly, organisms able to utilize this form of organic carbon should be favored. In springs with woody inputs, as seen in comparative studies of springs with and without plant biomass inputs. Archaea also play critical roles in the chemolithotrophic carbon fixation. The upper temperature limit for photosynthesis is pH dependent, as well as dependent on sulfide concentrations. In hot springs, photosynthesis occurs at up to 72 degrees Celsius in an alkaline geothermal environments. Above this threshold, non-phototrophic carbon fixation by chemolithoautotrophs provides critical inputs of organic carbon into thermal systems. To date, 2 CO2 fixation pathways, DC/4HB and 3HP/4HB, have been found only in hyperthermic archaea. In sites where DIC made up a large portion of total carbon, stable isotope probing with carbon-13 showed that DIC-derived microbial carbon driven in part by the Archaea community, was an important input across a range of conditions. A survey of hot springs in YNP that included 2 alkaline springs found autotrophic Archaea with DC/4HB pathway at high pH, but found no organisms with 3-HP/4-HB pathway. Methanogenesis. The methane cycle is broadly driven by members of the Archaea. Our understanding of the molecular pathways that underlie methanogenesis has expanded through genomics to reveal novel metabolisms, including hydrogen-dependent methylotrophic methanogenesis. Methanogens are widely distributed in anoxic environments, but the energetic costs of reducing CO2 increase at higher temperatures. Known methanogenic taxa are commonly detected in alkaline hot springs, but enrichments for methanogens from 2 circumneutral pH of 7.2 hot springs in Armenia found no evidence at 65 degrees Celsius. This suggests that evidence of activity from 65 degrees Celsius. This suggests that the poly-extreme conditions of both high pH and high temperature could limit the importance of methanogenesis in thermoalkaline sites, but known functional marker genes have been detected across a range of conditions. Targeted analysis of the methyl coenzyme M reductase MCRA gene across multiple hot springs in Tunisia, ranging from slightly acidic with a pH of 6.2 units to moderately alkaline with a pH of 8, successfully detected the MCRA only in alkaline hot springs with temperatures above 50 degrees Celsius. More recent metagenomic surveys found evidence of Archaea with MCRA across multiple hot springs in the Yunnan province in China. Closer examination of the alkaline pH of 7.6 to 9.6 units hot springs showed MAG from the Neza Archaeota, Vestra Archaeota, and Archaea globalis, Archaeota. In addition to widespread distribution, recent studies have revealed pathways for methanogenesis beyond the well-known hydrotrophic pathway, which is strictly anaerobic, in which CO2 formate or CO reduction is coupled with oxidation of hydrogen. Based on the presence of methyltransferase coronoid proteins and the absence of complete wood carbon dioxide reduction pathway, the Vestra Archaeota were classified as H2-dependent methylotrophic methanogens, whereas the other MAGs contain MCR genes associated with methanogenesis or methanotrophy, and encoded the conventional Wood-Ljungdahl pathway. Alkaline methanogens may also oxidize alkanes. Examination of the methyl coenzyme M reductase gene for anaerobic methane oxidation showed the potential for alkane oxidation, generally coupled to dissimilatory sulfate or sulfite reduction of nitrate reduction, was present across a range of phyla, including the Archaea globi, Hedysarchaeota, Nesaarchaeota, Archaeota, or Verstate Archaeota. Further analyses of MCR across archaeal lineages have identified MCR-like genes that drive the oxidation of short-chain alkanes, suggesting that genes linked to methanogenesis are involved in multiple metabolic pathways. The expanding number of taxa found to contain genes for methanogenesis beyond the hydrogenotrophic Archaeota and the emerging complexity of the methanogenesis molecular pathway suggest that additional surveys in understudied environments will further increase our understanding of substrate utilization by Archaea. Archaea and sulfur and nitrogen cycling. Archaea are known to have unique pathways for sulfur metabolism, coupled to methanogenesis, including respiratory reduction of the heterodisulfide of coenzyme M and coenzyme B in methanogens. Archaea also appear to have unique pathways for iron-sulfur cluster biogenesis, although the proteins required are still mostly unidentified. Sulfide-rich alkaline hot springs are thought to be relatively rare in most geothermal basins, and are found primarily in Iceland and India, but also in YNP, Papua New Guinea, and China. Alkaline hot springs not characterized as sulfidic can also show high levels of variation in sulfur compounds. For example, in the lower geyser basin of YNP, total sulfide in hot springs with a pH greater than that of 7.5 pH units in the Mounds region ranged from 3.65 to 19.5, but ranged below detection limit of 0.593 in the White Creek/Octopus Spring region. A survey of 4 hot springs representing the 4 main geochemical classifications in YNP alkaline chloride, mixed alkaline chloride sulfate, and travertine precipitation found that 80% contained sulfur, and even sites with low amounts of hydrogen sulfide. In contrast, sulfate concentrations in hot springs surveyed in New Zealand were high only in acidic sites less than 100 ppm. In a survey of metagenomes from YNP, genes from sulfur metabolism were generally more prevalent in acidic springs, but sulfur oxidation pathways have been documented in Archaea, which inhabit circumneutral to alkaline hot springs. Sulfur cycling appears to be absent in 2 superphyla, the DPANN, and the Asgard, which have no documented phyla with even partial metabolic pathways for sulfur cycling, as identified based on the absence of key genes. Ammonia oxidizing archaea, or AOA for short, tend to be found in acidic soils and sites, and have historically been thought to play critical roles in nitrogen cycle in acidic low nutrient content sites with at least minimal concentrations of sulfide. However, in soils, both the abundance and diversity of AOA are positively correlated with increasing pH, and adaptations to pH appear to drive niche specialization in AOA, suggesting that AOA plays key roles under alkaline conditions. Further, AOA have higher temperature optima and higher nitrification activity at high temperature than ammonia oxidizing bacteria, suggesting that they have competitive advantages under high temperature conditions. These general patterns hold in high temperature sites. In alkaline springs, pH roughly around 8. In Tengchong, a survey of AMOA genes that bacteria dominated at lower temperatures, less than that of 75 degrees Celsius, archaea were more abundant at temperatures of up to 90.8 degrees Celsius, and AOA were more active under high temperatures. Targeted sequencing of archaeal AMOA genes showed that AOA were present across a range of hot springs, including alkaline sites, pH of 7.6 to 9. AOA have also been isolated from non-acidic sites, including Nitrosocaldus yellowstonei from multiple hot springs in YNP, with pHs ranging from 6.2 to 9 units and temperatures ranging from 54 degrees Celsius to 90 degrees Celsius. And an obligately thermophilic ammonia oxidizer, Candidatus Nitrosocaldus islandicus, from a circumneutral hot spring in Iceland with a pH of roughly 6.5 and a temperature of 73 degrees Celsius. Based on high throughput sequencing of the 16S rRNA gene, Candidatus nitrosocaldus, a putative AOA, was found in thermoalkaline springs with pHs of 8.5 and a temperature of 75 degrees Celsius. In Y and P, and multiple OTUs similar to nitrosocoldus were found across a range of thermoalkaline sites, Figure 2. Again, as compared to acidic environments, relatively less known regarding the role nitrification by archaea in alkaline environments, but given the higher temperature and pH ranges of AOA, it is less likely that they play key roles in nitrogen-rich thermoalkaline springs. Archaea in thermoalkaline systems clearly play key roles in nutrient cycles, but in many cases, functions are inferred from organisms found in other conditions. Continued understanding of the true functional pathways as well as how organisms will interact requires additional focus on thermoalkaline sites. Even in YNP, arguably the best studied hydrothermal system, metagenomic sequencing has focused primarily on acidic and circumneutral hot springs. The limited coverage of high pH sites suggests that additional high coverage metagenomics could reveal additional insights into archaeal populations, communities, and metabolic pathways. Key taxa from thermoalkaline sites, metagenomes, and isolates. Alkaline hydrothermal systems are known to be reservoirs for novel uncultured archaea, as well as other microbial taxa with distinct metabolic pathways, and novel archaea are still being discovered in hot springs. Recent advances in sequencing technologies, including metagenomics and single-cell genomics, have uncovered many new archaeal lineages in extreme environments and have helped to improve our understanding of their ecology and function. Despite the limited focus of the thermoalkaline environments, metagenomic studies from other types of extreme environments have uncovered a broad range of phyla in the Archaea. Early applications of metagenomic sequences detected uncultivated archaeal lineages, Thaumarchaeota, Coarchaeota, and Igarchaeota. Analyses of these genomes have found that the newly described lineages often contain genes that code for novel metabolic pathways. By expanding environmental surveys to include a broader range of conditions, we would likely gain insights into the factors that structure the metabolic makeup of archaea, since geochemical parameters have always been shown to closely follow predicted functional pathways in archaeal metagenomes and other systems. To date, culturing efforts for archaea have yielded uneven results. Currently, the majority of isolates are methanogenic. Members of Euryarchaeotic and most phyla are known only from SAG and MAG reconstructions, but these reconstructions have yielded important insights into archaeal diversity and function. A number of recently described lineages have been found in thermoalkaline hot springs. Igarchaeota, TAC superphylum, are distributed globally and are found across a range of environmental conditions, but appear to be most prevalent at circumneutral to alkaline and moderately high temperatures, 75 degrees Celsius to 85 degrees Celsius. Increasing resolution in metagenomic and single-cell genomic studies has shown the potential for autotrophic mechanisms, dissimilatory sulfite reduction, and potentially carbon monoxide oxidation, as well as the ability to utilize a wide array of organic carbon substrates, including acetate, fatty acids, amino acids, and sugars, but are likely oligotrophic. Sequences with high similarity to Igarchaeota dominated the archaeal community in a thermoalkaline hot spring, but only in a single year. And in metagenomic surveys of an alkaline hot spring in Spain, Candidatus caldearchaeum subterraneum was the dominant archaeal organism detected and made up 4% of the classified sequences in the Phosmid library. Culturing from thermal systems has generally been skewed towards acidic or circumneutral sites. Archaea have been successfully isolated from alkaline geothermal systems, but as with the majority of archaeal isolates, have generally been organisms with previously characterized metabolic pathways. metagenome sequencing, assembly, and annotation using homology of observed protein coding genes against databases of better characterized isolates can be improved with the addition of single-cell genomics and metatranscriptomics to identify the poorly understood pathways of novel organisms. This vast volume of sequence data could in turn be used to inform new culturing strategies to isolate novel archaeal lineages from the environment. By informing cultivation techniques, for example, metagenomics and lipid profiles revealed novel MOA-like sequences that formed the basis of culturing efforts that led to successful growth of a novel lineage, the Candidatus nitrosocaldus yellowstonii, enriched from alkaline pH of 8.3 units, spring in Y and P. Additional nitrosocaldus isolates have been identified in a mildly alkaline hot spring, pH of 7 to 8, in Italy, and the circumneutral site in Iceland. Based on metagenome surveys, we found that a number of recently described taxa in the TAC superphylum have been documented in higher temperature systems under alkaline conditions, although some recently described lineages, such as the Marsarchaeota, have been found in acidic sites. Increasing attention to these systems will likely uncover additional taxonomic and functional diversity of Archaea, and also provide insights into the mechanisms of tolerance to polyextreme conditions. Figure 3. The number of thermal features, gray circle, in Y and P relative to studies of their microbial communities. Temperatures and pH data were downloaded from the Yellowstone National Park Research Coordination Network. Colored points represent the use of 16S sequencing with the Illumina MI sequence platform, 16S MI sequence, low coverage shotgun metagenomics, Sanger, 454, single end Illumina, low Cove MetaG, high coverage metagenomics, greater than 10 million reads per sample, primarily paired end Illumina, high Cove MetaG, and metatranscriptomics, MetaT. Data are taken from Coleman et al. 2016, Inskeep et al. 2013, Inskeep et al. 2010, McKay et al. 2019, Coleman et al. 2019, and additional data with no corresponding authorship in NCBI. The microbial community data are based off of publicly available sequence data from GenBank, NCBI, and that also had corresponding feature information, e.g. feature names or geochemical data. Due to limited reporting of pH and temperature or lack of named hot spring, this does not capture all hot springs surveyed to date, but those that have whole community microbial surveys paired with environmental measures. A number of sites were sampled in multiple studies using different methodologies, e.g. Monarch Geyser, Octopus Spring, and so have overlapping points. The dashed line shows the transition to alkaline sites. Marine thermoalkaline systems, serpentinizing sites. We have focused primarily terrestrially thermoalkaline systems, and hot springs in particular, but thermoalkaline systems can also be found in marine environments, particularly in serpentinizing locations. Serpentinizing systems have a high abundance of electron donors, including hydrogen and methane, but are often limited in electron acceptors. Although limited microbial surveys have been performed in these extreme systems, the geochemistry of alkaline hydrothermal vents have been well studied as the potential site for the origin of life, and have provided insights into the mechanistic underpinnings of critical geochemical cycles, both biotic and abiotic, including methanogenesis and iron cycling. Continental serpentine springs tend to be lower temperature, and microbial biomass in marine systems is often low, making comparisons with terrestrial hot springs difficult. In addition, unlike many thermoalkaline hot springs, the DIC and CO2 in these systems tends to be low, particularly in deep marine systems, potentially limiting chemolithoautotrophs. In the Lost City, targeted sequencing found taxa in the Crenoarchaeota and Euryarchaeota, as well as members of the ANME-1, and communities are often dominated by a single phylotype of methanogenic archaea. The lower diversity is likely due to the more extreme environmental conditions of deep sea environments, with higher temperatures and pH, as well as the absence of light, may represent a stronger environmental filter than seen in most thermoalkaline hot springs. Table 1. Archaea from the TACK superphylum and selected members of the DPANN, and Asgard, with representative genomes generated from metagenomic assembly or single-celled assembled genomes. The pH max represents the environmental measures of these sites examined. All sites had measures of greater than 65 degrees Celsius. The observed high pH of newly described phyla, such as the Mesarchaeota and Brachyarchaeota, could expand with additional sampling. Future directions for understanding archaea in thermoalkaline systems. A large number of archaeal lineages have not been cultured to date. The metabolic pathways inferred using genomics have provided critical insights into the niches occupied by archaea in thermoalkaline systems, but culturing these organisms can provide additional knowledge that may be gleaned only through the experimental manipulations. The vast volume of sequence data available can be used to inform new culturing strategies to isolate novel archaeal lineages from the environment. Using information from genome reconstruction, approaches to better design cultivation techniques are promising, and a number of these studies have been conducted in acidic and thermoalkaline environments. For these approaches to become more broadly feasible, additional genomic studies in the thermoalkaline environments must be undertaken to provide baseline information on taxonomy, metabolic pathways, and likely community interactions. The use of more recently developed omics approaches, such as metatranscriptomics, metaproteomics, and metametabolomics, have only recently been applied in situ thermoalkaline systems. Metagenomics provides a baseline for predicted function, but understanding realized function requires measures of transcription and post-translational products, including proteins and metabolites. Metatranscriptomic surveys allow for patterns of gene expression under varying environmental conditions to be quantified, as well as assisting in filling gaps in metagenome assemblies. Transcriptomic studies have also been used to quantify the likely functional pathway and niches of isolated organisms. For example, Beam et al. In 2016 generated a metatranscriptome for a novel Candidatus calditunius errorhumensis, could have named that something easier, and paired this data with extensive environmental measurements to better understand its metabolic adaptations. To our knowledge, metaproteomics have not been reported for hot spring environments, but analyses of individual proteins have been undertaken. These types of surveys, particularly when conducted across environmental gradients or with experimental manipulations, enhance our understanding of the functional roles filled by taxa in the archaea. Conclusions. Thermoalkaline sites represent extreme environments that are significantly understudied. Patterns found across the currently available literature reveal high pH habitats, likely host levels of uncharacterized archaeal diversity. Analyses of published studies indicate that temperature is the primary driver of archaeal abundance, diversity, and composition, but there is also evidence that pH, particularly above circumneutral, plays a critical role in determining the taxonomic and functional makeup of microbial communities. Additional studies that focus on high pH systems, particularly those that sample across broad geographic ranges with geochemical variability as well as temperature, have the potential to improve our understanding of which environmental parameters drive patterns of archaeal community assembly and function in extreme environments. Combined omics approaches that employ multiple methods, including metagenomic and metatranscriptomic surveys, which give a broad picture of the likely function, coupled with metaproteomic and metabolomics analyses, which provide a measure of the realized function within a given site, would be particularly powerful. These combined approaches will not only quantify the function of a given microbial community, but potentially provide a foundation for culturing of novel organisms. In addition, experimental manipulations could provide valuable insights into cellular mechanisms that drive archaeal communities, which would not only test fundamental hypotheses and theoretical ecology, but could also uncover novel metabolic pathways and identify enzymes with applications to biotechnology.